Welcome to today's lecture on the fascinating topic of interferon signaling in viral recognition and antiviral defense. In this lecture, we will explore the intricate mechanisms by which interferons play a vital role in detecting viral infections, inducing an antiviral state, and modulating immune responses to combat viral pathogens. We will also delve into the various evasion strategies employed by viruses to thwart interferon signaling. Lastly, we will discuss the clinical applications of interferon-based therapies. So, let's begin our exploration of this captivating field. During viral invasion, host cells activate pattern recognition receptors, PRRs, including Toll-like receptors, TLRs, and RIG-I-like receptors, RLRs, to detect viral nucleic acids or components. This leads to the production of type I and type III interferons that possess antiviral and immunomodulatory properties. Type I interferons have broader antiviral activity, while type III interferons mainly protect mucosal surfaces. Interferons, once secreted, bind to their receptors on target cells. Type I interferons interact with IFNR1/IFNR2, while type III interferons interact with IFNR1-IL10R2. These receptor engagements activate signaling pathways involving JAKs, STATs, and ISRs, resulting in the transcriptional activation of ISGs. Type III interferons use different STATs for signaling specificity. ISGs are genes activated by interferons that protect host cells against viruses. They produce proteins with antiviral functions, like breaking down viral RNA and stopping viral replication. Activation of ISGs depends on transcription factors like IRFs and NF-kappa B, which bind to specific DNA regions. Interferons exert their antiviral effects through the activation of antiviral enzymes, including protein kinase-R, PKR, 2,5-oligodinolate synthetase, OAS, and ribonuclease L-PKR inhibits viral protein synthesis by phosphorylating IF2-alpha, while OAS synthesizes 2,5-oligodinolates to activate ribonuclease L, which degrades viral RNAs. Interferons also enhance the expression of antiviral factors like MX proteins and viperin, collectively establishing an antiviral state in infected cells. Interferons interfere with various stages of the viral replication cycle to inhibit viral propagation. They elicit multiple inhibitory effects, such as blocking viral entry by downregulating viral receptors, inhibiting viral protein translation by targeting viral RNA, and impairing viral genome replication and assembly. Moreover, interferons enhance the expression of proteins involved in the degradation of viral components, such as proteasomes and autophagy-related proteins. These mechanisms collectively result in the prevention of viral spread and reduced viral pathogenesis. Interferons, in addition to their direct antiviral effects, play a pivotal role in shaping the host immune response to viral infections. They enhance the maturation and antigen-presenting capacity of dendritic cells, which are crucial for initiating an adaptive immune response. Interferons also stimulate the production of pro-inflammatory cytokines and chemokines, which recruit immune cells to the site of infection. Furthermore, interferons modulate the differentiation and function of T cells, B cells, and natural killer, NK, cells, strengthening the overall immune response. The crosstalk between interferon signaling and other immune pathways ensures an optimal antiviral defense mechanism. Interferons possess both autocrine and paracrine effects. Autocrine signaling refers to the ability of interferons to act on the same cells secreting them, enhancing their antiviral state. Paracrine signaling, on the other hand, involves the diffusion of interferons to neighboring cells, thereby providing protective antiviral effects to uninfected cells within close proximity. These dual effects ensure the amplification and spread of the antiviral response, enabling a robust defense against viral infections. While interferons play a critical role in antiviral defense, viruses have evolved various strategies to counteract interferon signaling and establish successful infections. Viral proteins, such as NS1, NS2, and NS3, antagonize different components of the interferon signaling pathway. They inhibit the phosphorylation and activation of STATs, disrupt the formation of ISGF3 complexes, or degrade IRFs, interfering with the induction of ISGs. Additionally, viruses may suppress the expression of interferon receptors, interfere with signaling pathway components, or produce viral factors that directly counteract interferon effects. These mechanisms collectively contribute to the ability of viruses to evade the host's interferon-mediated defenses. Interferon signaling does not exist in isolation but interacts with various other immune pathways to mount a coordinated defense against viral infections. For instance, interferons synergize with pro-inflammatory cytokines, such as tumor necrosis factor, TNF, and interleukin, IL-1, to enhance antiviral responses. Interferons also intersect with the innate immune system through interactions with natural killer cells and macrophages, further strengthening the host's ability to combat viral pathogens. The delicate balance between these interconnections fine-tunes the immune response and ensures effective viral clearance. Although both type I and type 3 interferons contribute to antiviral defense, they exhibit some distinct properties and roles. Type I interferons are rapidly induced during viral infections and provide immediate protection against a wide range of viruses. They act systemically, diffusing through the bloodstream to reach target cells. In contrast, type 3 interferons are predominantly expressed at mucosal surfaces, orchestrating localized antiviral responses. They play a crucial role in mucosal immunity and are particularly effective against viruses that primarily infect epithelial cells, providing an additional layer of defense at the site of viral entry. Viruses have evolved sophisticated strategies to evade interferon signaling, enabling them to establish persistent infections. Some viruses, such as hepatitis C virus, HCV, and human immunodeficiency virus, HIV, directly target specific components of the interferon pathway to subvert host defenses. For example, HCV inhibits the phosphorylation and nuclear translocation of STAT1, impairing interferon-induced gene expression. HIV incorporates proteins, like VIF and VPR, that directly antagonize the function of specific ISGs, limiting their antiviral effects. Interferons have been harnessed for therapeutic use to combat viral infections, including hepatitis, human papillomavirus, and certain viral-induced cancers. These treatments aim to boost the body's immune response while suppressing viral replication. However, challenges like side effects and the need for improved delivery methods and targeted approaches persist in the clinical use of interferons. In summary, interferons play a crucial role in combating viral infections by activating a variety of antiviral mechanisms. However, viruses have evolved ways to evade interferon signaling. Understanding this interplay is vital for developing effective antiviral therapies and improving patient outcomes.